Hello everyone, welcome back to the session 7 of Principles of Inheritance and Variation. So how was your Sunday? I hope all of you got some time to either read your NCERT or practice some questions from this chapter. I see that a lot of you are already here. So hello everyone, I'll just wait a couple of seconds. And by that time, all of you can tell me if you were able to read the NCERT or if you were able to do some of the questions, from the model test papers in the portal. So just write it down till we wait like a couple of seconds for other people also to arrive. So quickly let me know, did you get some time to read either the NCERT or do some questions? Okay, even if you did not get the time, for this Sunday, try to do it. Because see, we are doing all these online lectures for you. It will really help you, if you do it simultaneously, It will kind of finish your one chapter for both your board exams and both your NEET exams. For both of them, it will help you. Okay, so I see that a lot of you are already here. So let's begin. So this is the overview of the chapter, and last week we have seen a lot of things. We have learned a lot of new interesting things, right? And the last thing we discussed before signing off for the weekend was genetic disorders. So quickly we will revise what all genetic disorders we have seen. Okay, so we have seen this table multiple times, and try to make a diagram of this table in your mind every time, so then you will be very clear. Okay, and we have tried to cover small sections in one lecture. So just try to revise this table and try to revise simultaneously all the genetic disorders, because this will contribute to a lot of questions in your entrance exams. So genetic disorders are chromosomal disorders and Mendelian disorders. Chromosomal disorders where the disorder is because of the chromosomes. And again, it can be either polyploidy or aneuploidy. What was polyploidy? Polyploidy was when the whole set of chromosome is changed. Aneuploidy when only one or more chromosomes are changed. And for aneuploidy, again, we saw two types. We saw aneuploidy of the sex chromosome and we saw aneuploidy of the autosomes. So for the sex chromosomes, we saw Turner syndrome and Klinefelter syndrome. I hope you remember. While I am saying, just try to also remember all those things, okay? And for autosomes, we saw Down syndrome, trisomy of chromosome number 21. Then we also discussed about the Mendelian disorders or traits. We said that these are not always, disorders. These can be also some traits. And then they are again of two types, autosomal and sex-linked, and we focused only on autosomals for now. And in autosomal, again, it was of two types. One was polygenic and one was monogenic. What is a polygenic trait? Multiple genes controlling one character. For example, skin color. Okay, there were 3 different genes that were involved in determining the skin color. That is why we have such varied degrees of, colors. Then we saw the monogenic traits where only one gene is involved. We saw sickle cell anemia. Sickle cell anemia, what was happening there? The beta globulin chain was defective because of point mutation. And I hope you remember the question that we did. In the normal, it is glutamate. In the mutant, it is valine. So don't mix it, okay? Then we saw thalassemia, both alpha and beta thalassemia, which is again due to reduced synthesis of the beta globulin chain. Then we saw one of the most important things, and you don't have to be confused. Again, we saw sex-limited trait. It is a sex-limited trait, but it is an autosomal disorder or autosomal trait. Reason being because the genes are present on autosomes, but it is influenced by the sex. Okay, so we saw sex-limited trait. So remember Adam's apple. Adam had an apple. It was stuck in his throat. I don't have an Adam apple, so I cannot show you. But all the guys out there, they have this bulge here. Then milk production in the breast. These are all sex limited traits because only one, either the male or female, will have it. Next was sex influenced trait. Again, we saw the example of baldness, and remember, your grandparents are generally the grandfathers are bald, but the grandmother will still have some hair. This is because it is a sex influenced trait. Okay, good. Then now we again continue with the monogenic. Okay, and in monogenic, now we talk about multiple allele traits. Again, this is not a disorder, this is a normal condition. Okay, so in such traits, the concerned genes, instead of 2 alleles, one dominant and one recessive, have 3 or more alleles. For example, the ABO blood group system, and you will be like, what? How can it have 3 alleles? You remember when we talked about genes and chromosomes, we said these are the chromosomes and these are 2 homologous chromosomes, and these 2 will form, the 2 alleles will form a gene. So now why are there 3 alleles? So this is very important. Listen to me very carefully. In the ABO blood group system, there are 3 alleles in the population. Okay, but every individual will still have 2 alleles. Okay, every individual will have 2 alleles because 2 alleles always form one gene. Alleles are 2 different molecular forms of a gene. So that is why it is called as multiple alleles. So don't confuse yourself. In the ABO blood group system, we have 3 alleles in the entire population. By entire population mean all the humans. Okay. Then there will be different combinations of these 3 alleles, but every individual, for example, I will have only 2 of those 3 alleles. You will have only 2 of those 3 alleles, but they can be different. It will become more clear. So we all know, I think this we have studied since a lot of years now, that there are 4 types of blood groups: blood group A, blood group B, blood group O, and blood group AB. And then they follow a mechanism that is called as codominance. Now we have always said that one allele is dominant, the other is recessive. This is again, if you remember, when we were studying about linkage, when we were studying about incomplete dominance, when we were studying about pleiotropy, we also studied a little bit about codominance. What are all these? These are deviations to the Mendel's law. Okay. So what happens in case of codominance? Both alleles of a gene are equally dominant here. For example, the dominant character is not able to suppress the recessive character, and thus both the characters appear side by side in F1 hybrid. So F1 generation represents both the parents. Now if you go back to this example that I always give, mother is dominant, father is recessive. Now mom wants to cook sambar, but dad wants potato. Mom wants lentils, dad wants potatoes. So in a normal Mendelian situation, we will cook lentils because mom is the dominant gene. In such a situation, we will cook both. We will cook lentils and potatoes. So the F1 progeny will get to eat both. Okay, just remember this example. In case of codominance, they are both expressed. And how? We will see. So we will see the genetic basis of this ABO blood group system. There are 3 alleles in this system, and which are those 3 alleles now? A, B, O, present on the longer arm of 9th chromosome. Okay, a chromosome always is represented like this. Okay, there is a short arm and there is a long arm. The short arm is the P arm, the long arm is the Q arm. Okay, that is why it is present on the longer arm of 9th chromosome. And remember this, you remember last time we practiced a question about phenylketonuria. How important is this location? So you have to remember the genes are present on the 9th chromosome. If you did not write it down, in the last lecture, start writing it down now. Okay, the blood group alleles are present on the longer arm of 9th chromosome. And therefore, it is an example of multiple alleles. Okay, because there are 3 different alleles, A, B and O. Then both alleles A and B are dominant over O, showing of character. Thus, allele O is recessive to both A and B. Now, mom wants to cook lentils, dad wants to cook potato, the baby wants pizza. Nobody is going to listen to the baby, okay? Either they will cook lentils or they will cook potatoes or they will cook both. But in no situation, they will make pizza. They will make pizza only in the situation when both the parents also want pizza. Okay, is it getting clear? That is how you have to remember this, that A and B are dominant. They are co-dominant. When they are present in the F1, they are present together. O is the recessive one. Okay. And with this table, I think things will become crystal clear. Now, so blood type A can be represented as either IA, IO or IA, IA. So this IA is what? It is the homozygous situation. Okay, in any case, when they are homozygous situation, whatever is there will be expressed. So the blood group will be A. So here the blood type is given A. The second situation is when it is IA, IO. And most of the times it can also be represented as small I. Okay, so IO is equal to small I. So don't get confused, if I start writing small I or if in questions you see it as small I. It is up to you. You can choose how you want to write it, either IO or I, small I. In such a situation also now, when this is the genotype, now what is happening here? This is the dominant one. This is the mom that wants to cook lentils. Here is the pizza. No, in the end you will cook lentils. A is dominant over O. Okay, that is why the blood type will be A. Then next is B. Again, either it will be IB, IO, and in this case again B is dominant because both IA and IB are dominant and IO is the recessive. Okay, these are both the dominant and IO is the recessive one. So then again it will be B blood type in both these genotypes. Now comes the interesting part. Why do we say it is codominance? Now you see AB, one allele is A, other allele is B, and that is why the blood group is AB. That is why it is codominance. Both the things together, nothing is suppressing each other and both of them are equally expressed. That is why the blood group is AB. And the only condition when the blood group will be O, when it is IO, IO as normal in the recessive situation. Is this thing clear? Is this concept of codominance clear? Is it clear that whenever it is IA with O, it is A? Whenever it is IB with O, it is B. But when it is IA, IB, it is codominant, it is AB. And in only the situation where it is IO, IO is the O. Is it clear? So quickly let me know and then we proceed. If there is any question, then let me know. So I'm waiting till all of you give me thumbs ups. Good, good. Then let's proceed. Okay. Now we will do one question. What can be the blood groups of the children if the blood group of the parents are A and B? And one thing that you have to always take into consideration whenever you have such kinds of questions. So this is not the question from previous year papers, okay? I just took it from some book so that we can understand how this thing works, okay? How this process works. So what can be the blood groups of the children if the blood groups of parents are A and B? And to find out the possible genotypes in children, the genotypes of the parents are always considered to be heterozygous until and unless it is specified, okay? Until and unless they say it is a homozygous A, you will always consider that A means IAIO. Until and unless it is specified that the parent is a homozygous A. And that is how you are going to solve all such kinds of questions. So I will help you to solve this question. If any one of you can quickly solve this and give me the answer, perfect. But let's just see how this is done. So what can be the blood groups of the children if the blood group of the parents are A and B? So for example, let's say the first parent is IAIO. Again, we take heterozygous because we have to see the blood groups of all the children. And then B will be IBIO. And then you will do the normal cross. IAIB, IAIO, IBIO, IOIO. So these are the 4 possible genotypes. And then what are the phenotypes? AB. This one is AB. This is A because IA is dominant over IO. This one is B because IB is dominant over IO, and this one is O. So these are the 4 blood group types that are possible. Perfect. A lot of you have answered. See, some of you answered just A, B and O. Some of you have given the right answer for all. So these are the 4 possibilities, you know. All of these 4 possibilities are possible. Possibilities are possible. All of these are possible. Okay, when the parent is heterozygous A and heterozygous B. That is why I told you, you know, when they are asking you a question, what can be the blood groups of children if the blood group of the parents are A and B, you have to always keep in mind that you take heterozygous parents. Okay, for example, if you quickly take homozygous parents, IAIA cross with IB, then the only possibility is IAIB. That is AB blood group. So don't do that. That is why this is very important to remember. If they have not mentioned it in the question, always consider it as heterozygous. Is it clear for all those who gave me the answer just A or B or AB or O? Is it clear? You see, you have to do such kinds of cross. This we have learnt multiple times, no? We have done it a lot of times. So is it clear that whenever they haven't mentioned in the question, you have to consider that they are talking about heterozygous. Whether they say parents, whether they say children, whatever they say, you have to consider that it is heterozygous until and unless they mention it is homozygous A blood group. Good. Then let's proceed. Okay. So this question you are going to solve for me. Again, it is not a previous year question, but this will help us to understand the concept. If the blood group of mother and the child is A and B respectively, then the person of which blood group can be the father of this child? Just write down, you know, the genotype of the mother, write down the genotype of the child, you will automatically get the blood group of the father. Tell me both what possible genotypes are possible, what are the possibilities of genotype and what will be the phenotype. It can take a while. If you are not able to do it, we will do it again together, but try. It's very easy if you just write down the genotype of the mother, write down the genotype of the child and then try to see, you know, what is missing and what can come from the father. So I will wait till all of you give me the answer. Just write down, you know, try to write it down and do it. The best way to do all these numericals of genetics is to always write it down. Don't try to do it in your mind, you know. It will just take you 3-4 seconds extra, maximum 30 seconds extra, but it will never give you negative marking. Okay, so just try. Okay, I got one answer, but I will still wait till I get all the answers. I start getting some more answers, but I want little more. You always want more. Okay, good. So, let's do it again together. If the blood group of the mother and the child is A and B respectively. So, here is the mother. What is the blood group of the mother? A. So, it is IAIO. And then the baby that is born here has this is A blood group, the baby has the B blood group. Okay, so what are the possibilities of the B blood group? The possibilities are either it will be IB or it will be IBIO. Okay, that is only the case when it will be B blood group. Now, if you look at this one, you know, that is definitely not possible because it means that this came from father, this came from the mother, and mother does not have this. So, this is not possible at all. Okay. So, the only possibility for the genotype of the child is this, and where from the mother this IO has come. Now, the father can give IB, which means it should definitely have B. And what are the genotypes that will have B? IB, IBIO, or IAIB. Okay, if it is this two, then it is B blood group, and if it is this, then it is AB blood group. So, these are the possibilities for the father. And most of you, actually all of you, have answered the right answer. Okay, it cannot be A. The father cannot be A because here we know very clearly that if the baby is B, it definitely has one IB. Okay, and from the mother, the baby took IO, so this allele IB should be definitely present on the father. In case of A blood group is possible only when it is IAIA or if it is IAIO. There is no B here. B is not dominant or recessive. Therefore, A blood group of the father cannot be possible. Good. I see all of you have answered the right answer. Some of you said A also somewhere. Okay. So, just if there is a little confusion, then it should be clear now. Good. Very good. Then let's proceed. So, again coming back to the overview of the chapter, we have seen a lot of genetic disorders. Okay, and then we will continue with these now. So, we have already seen Mendelian disorders and traits. Okay, we have seen autosomal, polygenic, monogenic, and the last one in monogenic was multiple allelism. And again, you know, just revise the fact that they are all called as monogenic because there is only one gene. On the 9th chromosome are the genes present for multiple allelism or for blood group ABO. Only one gene is involved. Okay. Now we come to the next Mendelian disorders. We have already discussed the autosomes. Now we come to the sex-linked. Remember, again I am stressing, don't get confused. Don't confuse this with sex-limited or sex-influenced. And then again in sex-linked, there is X-linked and Y-linked. So, let's proceed. So, what are sex-linked traits? So, they are sex chromosomal traits, but they are still Mendelian traits. Why? Because the genes are present on the sex chromosomes. Okay, but they do follow this Mendelian pattern. It is not about gametes, it is about the genes. Okay, so such traits are governed by genes present either on the X chromosome or on the Y chromosome, and more common are generally the ones on the X chromosome from the point of view of your board examination and your NEET examination. So X-linked or sex-linked traits. These traits are governed by the genes which are present on that part of X chromosome which does not have its homologue in the Y chromosome. Thus, the X-linked genes are sex-linked genes. Okay, we always talk about homologous chromosomes, but in case of XY individual, what happens is, some part of the X chromosome have corresponding genes on the Y chromosome. But the sex-linked traits talk about only those genes that do not have a homologue on the Y chromosome. Okay, this should be very clear. So all the genes of these sex chromosomal traits or sex-linked Mendelian traits will be on the X chromosome when we talk about the X-linked traits. If we talk about the Y-linked traits, then it is definitely present on the Y chromosome. Good. So the first trait or the first disease that we are going to talk about is hemophilia. Again, there are some things that you have to remember. So it is also known as bleeder's disease, and trust me, I have seen this question from the previous year questions. We are not practicing it today, but I have seen it. Which one of the following is the bleeder's disease? So just remember, okay, don't think that anything that is there in these presentations that is there in your NCERT you can skip. No, when you are doing one chapter, do it everything, no? Then that chapter will be holistically over for you, for the point of view of your boards and from the point of view of NEET. So it is also called as bleeder's disease as blood clotting in affected person is delayed. So what happens, whenever I chop vegetables, I'm very bad at chopping vegetables, but I have to do that. I live alone, I have to cook every day. And most of the time it happens while chopping, I'll get a cut. Oof. And then it will bleed. And then what I'll do, I'll just take a tissue paper, I'll hold it or I'll put it under the running water. And after a while, it will be fine. It will be perfect. Why? Because the blood clots. But in people with hemophilia, this is not the case. There will be very delayed blood clotting. They will lose a lot of blood before it starts clotting, and sometimes it doesn't clot at all. Okay, so that is what hemophilia is. So it is mainly of two types, hemophilia A and hemophilia B. And again, something that you have to remember. Hemophilia A is more common and develops due to absence of blood clotting factor 8 AHG, anti-hemophilic globulin. You must have studied in your 11th standard, right? Then you studied physiology. You would have studied that pathway of clotting. If you don't remember, at some point, Concept Tree will also do lectures for 11th standard. Join them, revise your 11th class. Okay, but here then try to remember. Hemophilia A, blood clotting factor 8, AHG. Hemophilia B, which is less common, it occurs in 20%, develops due to absence of blood clotting factor 9, PTC, plasma thromboplastin cofactor. Remember these. And what is the best way to remember these? As I always say, write it down. Write it down on this sheet of paper next to you where you are writing the names of the scientists, where you are writing which disease is associated with which chromosome number. Write it down there. It will be very helpful. Okay. Then hemophilia B is also called as Christmas disease as it was first discovered in a Christian family. Okay. So bleeder's disease will be both hemophilia A, hemophilia B, and hemophilia B is also called as Christmas disease. I haven't seen this question, but they have the potential to ask this as well. Okay, so just remember this. It was first discovered in some Christian family. Probably, I don't know, maybe one of them cut something and then they discovered when they ran to the hospital. Maybe. Okay, so then let's proceed. Good. So then we talk about why it is a Mendelian disorder. It is again sex-linked Mendelian disorder and the genes are on X chromosome. So these are the genotypes for hemophilia in males and females. For example, in case of males, XHY is normal, and again capital H, small h. Capital will be the dominant, small will be the recessive, right? So in case of males, it is XHY, that is normal, and X small hy is hemophilic or diseased. Now let us look what happens in females. In females, we have two X chromosomes. So in males, XH, capital H and small h, okay. Capital H will be dominant thing, small h is recessive. When the dominant is present, it is normal. When recessive is present, it is diseased. So in case of females, capital H is homozygous, it is normal. Capital H, small h, again heterozygous, normal. Why? Because dominant will mask the expression of recessive. And then XH will be homozygous disease. Now you have to write it down in the chat box. Is hemophilia a dominant sex-linked disorder or a recessive sex-linked disorder? Just see what is written in front of you. It's a very easy question. If it is sex-linked dominant disorder or if it is a sex-linked recessive disorder. Quickly let me know. Just write down whatever you feel, okay? Just see what I have told you in males and what I have told you in females. Good. All of you write it down. It's okay, even like if you don't know, just say I don't know, okay? That is how we will learn. Good. Perfect. Most of you are writing it recessive and that is the right answer. See, when it is X small h, only then. So in case of females, both the alleles have to be recessive to get the disease. But what happens in case of male, even if one of the X chromosome is recessive, it will be hemophilic. Why? Because in case of males, there is only one X chromosome. Okay, in case of males, there is only one X chromosome. Therefore, if that is recessive, there will be still disease, because when recessive is present alone, if small h is present, nothing is there to mask its expression and therefore it will be expressed. And the same thing happens, in case of males, because we also discussed that these are the genes on X chromosome that do not have a homologue on Y chromosome. So Y chromosome is perfectly fine, but the only X chromosome in case of males is recessive and therefore it will be hemophilic. Is this concept also clear, guys? Like why in case of just males, presence of only one X chromosome that is recessive will result in the disease. Because there is nothing to mask the expression of small h. Something is dominant or recessive only if it is present in a pair. Here only, h will mask. If small h is present on its own, doesn't matter, if dad is alone one day at home because mom has gone to the relatives, he will cook potatoes. He will not cook like lentils and potatoes. He will just do what he wants to do. So that is why small h, when it is present alone, will do whatever small h wants to do, and that is hemophilia. Is it clear? Let me know if it is clear, or should I repeat it again? Because this will help us tomorrow when we are solving the pedigree analysis. One of the areas that people are most scared of. I was, but we'll do it again in a very interesting way. So let me know if this concept is clear, that in case of males, you need only one defective X chromosome. In case of females, you see, in the heterozygous condition, she is normal, because here there is a capital H to mask the small h. Good, perfect. And then this you have to remember, when you are solving problems related to these sex-linked inheritance, the normal will be represented as X only. So for example, if you talk about the homozygous normal, it will be XX. If you talk about this heterozygous normal, it will be X small h. Okay, so that is generally how you can solve the question, and sometimes if there are in questions, that is how you have to take care. Then let's proceed. You want to try this yourself? Just try it yourself. Try it yourself. I will give you like a couple of minutes. If the father is hemophilic, or I will write you the genotypes, okay? You do the cross and then tell me. If the father is hemophilic and mother is homozygous normal, predict the genotypes of children and grandchildren. So you just tell me the genotype of the children. Okay, let's not talk about the grandchildren right now. Just tell me what will be for the children. The father is hemophilic and mother is homozygous normal. Try to solve it yourself and then we will do it together. If you cannot, just write it down. It was not possible. These questions take, initially they will take quite some time for you to solve, but slowly when you practice a lot of them, you will get used to it. So just tell me for the children right now and then we will talk about the grandchildren. Okay. So I start getting the answers, but we'll see if this pen works. Yeah. Okay. So father is hemophilic. Okay. So what will be the genotype of the father? Good. So I don't know what 1:1 ratio means. You have to write it down. Homozygous, heterozygous, normal, hemophilic. Okay. That is how I will get to know. But okay, you have attempted it. Very good. And I'm sure the answer you've given is also right. So if the father is hemophilic, then what will be the genotype? XHY. And mother is homozygous normal, X small h. Okay, so these are the genotypes and then again you will do the cross. So, these are what are the possibilities there. Okay, no, sorry, I am sorry. So, the mother is homozygous normal. She is not heterozygous normal. Just delete it. And this pen doesn't work. Okay, I think I'm really distracted because of this technical issue today and I'm really sorry about that, guys. Okay. So I see most of you already are writing the correct answer, which is what? You will observe one genotype will be XH small h, X small h, X, that is a carrier. Second is XHY, that is a son who has the disease. Third is XX, that is normal, and fourth is XY, again normal son. Okay, now good, just forget about that. Okay, so XHY crossed with XX. So this is what all you get here. Good. So now what you get? You get the normal son and you get carrier females. Okay. You get the normal son and you get the carrier females. Now what is the next question? Now you have to predict the genotype of the grandchildren. And when you will get such kinds of questions, and whenever you get such kinds of question, what is the next step? You have to take the carrier female here and then you have to cross it with a normal male. I'm sorry, I don't know what happens sometimes, you know, this pen doesn't work. Okay, so what is the next step is that you will take this female, you know, XXH, the carrier female you will take, and then you will cross it with a normal male, XY. Just try to do that. Try to do the second cross for the grandparents. I see already some of you are answering for the grandchildren and I am lagging behind. You imagine the teacher is lagging behind today. Okay, so here we go. Okay, and we are back with this pen. Okay, I will really buy a new pen, I promise. This won't happen again. Okay, good. So we already saw that in children there was this carrier female, and then when we cross this carrier female with the normal male, now we get again 4 different types. One is what? One is normal female, one is normal male. Then one is normal carrier female and hemophilic male. You see, these are the possible genotypes. One hemophilic male, heterozygous normal, normal male, normal female. Okay, in all this mess, are the crosses clear? You have to tell me, otherwise tomorrow I will start the lecture with this question again and I will try to buy the new pen. Yeah, I see the answers are coming. Okay. So one thing, you know, that we have to understand from what we did just now in all this mess is that whenever we have a carrier female and a normal male, we will have an affected male. Half of the males will be affected and half of the males will be normal. Okay, this we have to really understand. This is because one of the X chromosome will definitely come from the mother, and therefore when it splits into half of them will be normal and half of them will be affected, because the Y chromosome from the male is always coming to the son. It will become more clear soon, and tomorrow I will repeat it again. I am really sorry. Okay. Then let's go to the second trait, that is color blindness. So color blind person cannot distinguish between red and green colors. Okay, this is the problem. They cannot distinguish between colors. That is why, you know, sometimes they say that don't use such colors in your presentation, and I am always using red. I hope none of you are colorblind, but in this case, you know, it is not a problem. When does the problem occur? You know, such persons cannot be appointed in traffic police or cannot be issued driving license. Why? Because it is red, yellow, green. Red stop, yellow stop, green go. Not everybody follows that, but that is the real rule, okay? And that is the reason, because they cannot distinguish, you know, between red and green colors, and that is why they cannot be as a traffic police and they cannot be given driving license. And it is the most common sex-linked disorders, and they are also again expressed in the recessive condition, similarly like hemophilia. And here again, you know, normal will be X, and if it is XC, then this is C, that is for color blindness, and such a female will be again normal. This female will be affected. Okay, and if the male has only one, again it will be affected. And again, similarly like before, you know, the normal will be X and not capital XC. Good. So, then let's try to do this question again. Like it's a similar question, you know. Miss Pooja, whose father is color blind, marries Mr. Rakesh, whose mother is color blind. Now, don't get confused, okay? Just write it down. Miss Pooja and Mr. Rakesh. So, you want to try it yourself? Just try it yourself. Kavya, you asked me a question. I will take that question tomorrow. Okay, tomorrow's lecture we will start again with the same example, and then I will be also able to, you know, explain you properly, because now it's really a mess. So Kavya asked, how about the genotypic ratio? We'll discuss this question in very much detail, okay? The previous one. Tomorrow we will start our lecture with that. So just try to see if you can do this. I will try simultaneously with you. Okay. Just try doing it yourself. Okay. So here is Miss Pooja, whose father is color blind. So the father will have XCY, and she will definitely take one of the X chromosome from the father because she is a female. Therefore, she will be a carrier. Okay. And Mr. Rakesh, whose mother is color blind. So her mother has XC. So he will definitely take this X, and therefore his genotype will be XCY. And then again we are going to cross. Okay, so these are the possible progenies that are there. So you have to predict the probability of color blindness if the child is a son. So we see here, yeah. So some of you have already answered. So it is good. So you see here, out of the two sons, one of the son is color blind. Okay. And out of the two daughters, one of the daughter is color blind. The other one is normal. Although she is a carrier, but she is normal. Therefore, the ratio if the child is a son. So, whenever you calculate this ratio, again, this can be sometimes tricky. Whenever you calculate, you know, they are asking you when the child is a son. So, you have to consider only son. So, two sons will be, like ideally in the ratio, there will be two sons. Half of them are normal and half of them are color blind. Therefore, the ratio is 50%. Don't try to say that the ratio is 25% because there are four possible progeny. Read the question properly, okay? They say when the child is a son. So, you have to consider only these two possibilities in this case, and then the ratio is 50%. The probability of color blindness then is 50%. And if the child is a daughter, again, just talk about the daughters. And if you talk about the daughter, again, the ratio is 50%. And I see some of you were able to answer, right? But for others, is it clear? Is it clear? Is this question clear? How you will go about solving it, or it became a complete mess? Just let me know. Because this is important. One normal male, since females are carriers. No, Lakshmi, here, you know, when you do a cross, you see, that is why I always say, you know, you have to always do the cross and see. So, this one, XC, the one that will take one defective allele from the mother and one defected allele from the father will be colorblind. So is it clear, Lakshmi, for you? Because I think for B you are saying 0. That is not the case. That is why I always say write it down. You know, such questions you have to write down. I'm still waiting for Lakshmi to tell me, because she said that B is 0 since females are carriers. They are, yes. But in this case, the father is defected and the mother itself is a carrier. So, if the female takes both the defected alleles, you know, one from the mother and one from the father, then it will be defected. So, I am hoping it is clear. So, then we proceed. So, next is deficiency of G6PD. This is again sex-linked Mendelian trait. The enzyme glucose 6-phosphate dehydrogenase is present in RBC for minor glycolysis. You know what is glycolysis, right? The process by which energy is produced, glucose is utilized. So, there is another process of minor glycolysis taking place in RBCs, and glucose 6-phosphate dehydrogenase is responsible for this. The absence of this enzyme is a sex-linked recessive trait. Okay, and the person deficient in this enzyme cannot have plasmodium in their RBC. And if you remember from the last lecture, you remember we discussed about sickle cell anemia, we discussed about thalassemia, and both these are protected against plasmodium that causes malaria because the shape of RBC changes. And in this case also the plasmodium cannot enter the RBC because there will be an effect on the shape of the RBC. Okay, and hence they cannot suffer from malaria. But if such people are given anti-malarial drugs, the RBCs, because they are fragile, can rupture and they can cause severe anemia. And this can be fatal for the patients. Okay, why this happens, you know, we don't know. If a person who has deficiency of glucose 6-phosphate dehydrogenase, what will happen? Plasmodium cannot enter the RBCs. But for instance, by mistake, if they take some anti-malarial drugs, their RBCs will become fragile and they will rupture, leading to severe anemia, and this can be fatal for the patient. Therefore, the people who have deficiency of G6PDH, they should be very careful that they should never take anti-malarial drugs. Okay, generally it won't happen, because why would you take an anti-malarial drug when you cannot get malaria, right? They are anyways protected against malaria. But sometimes it can happen. Like nowadays, we all know that in this situation, you know, they are trying different types of medicines, you know, some medicines that are for a different disease. They are trying to treat this virus for that. So these patients should definitely not do the trial version. That is why it can be really fatal. And the percentage of G6PDH deficient person is more in South Africa, a malaria prone area. And we saw, okay, sickle cell anemia is also not completely eliminated in that population, because these diseases, although they are diseases, but they give them an additional advantage, because South Africa is much more prone to malaria. The plasmodium is a lot there. Is it clear? This concept of G6PD, glucose 6-phosphate dehydrogenase. If it's clear, now you have to write in the chat box where are the genes, which chromosome number or which chromosome has the genes for G6PDH. Write it down in the chat box. We have discussed this before. If you remember very well, if you remember the concept, you can answer this question. Where are the genes for G6PD present? Glucose 6-phosphate dehydrogenase. So just keep in your mind, you know, South Africa is a really malaria prone area, okay, and that is why a lot of people will be heterozygous for sickle cell anemia. They will also have deficiency of G6PD. Coming back to, if you don't know the answer, just write down I don't know, but just give me some response so that I know that we can proceed or not. quickly tell me, where are the genes for glucose 6-phosphate dehydrogenase enzyme present? Okay, I don't know if this is the lag or none of you knows the answer. Just write down that you don't know if you don't know. Okay, I start getting the answers now. Good. Write down. Just write down that if you don't know, you don't know. See, some people are writing. Good, good. Keep on writing. Everybody has to write. I will not proceed till everybody of you writes, and then your dinner will be delayed. Good. I just have 3 answers. Maybe there is a lag. So let's just wait and see. It's very interesting answer. But good. I'm happy that you're replying. I think others have already left for dinner, or there is a lag. Yeah. Okay. 4 answers I have received. I want more. You always want more in your life. Okay. So 9th chromosome, 11th chromosome, is it 11th chromosome, 11th chromosome? What is deficiency of G6PD? What is the title of this slide? Sex-linked Mendelian traits. X-linked. The absence of this enzyme is a sex-linked recessive traits. Where are the genes present? They are present on the X chromosome. Why it has to be the 11th chromosome? Why it has to be the 9th chromosome? It is the sex chromosome and it is the X chromosome. That's why you have to listen, very carefully. Okay? It is the X chromosome. The genes are present on the X chromosome. Is it funny? You see, that is why you don't have to use extra brain. You just have to see what was there in the slide. You have to just read the heading. It is an X-linked disorder. The absence of this enzyme is a sex-linked recessive trait. How many sex chromosomes we have? Just XX and XY, and it is an X-linked trait. So genes are present on the X chromosome. Yeah, this is that oops moment, But it's okay. That's why we are here. We are here to learn. Okay, but is it clear now? Okay, so the genes are present on the X chromosome. The genes for haemophilia, genes for color blindness, deficiency, the genes for glucose 6-phosphate dehydrogenase, all are present on the X chromosome. Good. Then let's proceed. Question time. G6PD dehydrogenase deficiency is associated with hemolysis of. Very easy and straightforward question, no? Leukocytes, lymphocytes, platelets or RBCs. What is the right answer? You see, now a lot of you, I see these laughing smileys. It's okay guys, that is why I say, we are here. We are here to learn. Good. I start getting the answers for this question. It's a very straightforward question, no? But it will be asked again. This is not my question, not taken from any book. It is directly taken from the previous year question papers. Good. So I get all right answers and the right answer is D, RBCs. Good. Now we'll quickly discuss, this is something very small, Y-linked or Hollandric traits. You have to remember this word, Hollandric. Okay, so the genes that are present on the Y chromosomes are called as Hollandric genes, and the diseases that are linked with these genes are called as Hollandric traits or the traits, okay. They are governed by genes present on the Y chromosome, and such traits pass from father to all his sons. Obviously, no, because the sons will take the Y chromosome from their father, so it will be from father to son, father to son, father to son. For example, hypertrichosis, and remember this, direct question will be asked. What is hypertrichosis? Excessive hair on pinna. And when we were discussing about Down syndrome, I told you that pinna is lowered in case of people with Down syndrome. What is pinna? Pinna is this outer ear, And for all the guys out there, the boys out there, you have to see, if you have excessive hair growth here, definitely your father also has excessive. See, some amount of hair is always present. But when it is too much, that is the reason, because you got it from your father. Females will never have that. Females will never get a Y chromosome from their father, so they will not have that. So what is important from this slide is, why we included this slide is, you have to know that the genes on Y chromosomes are called as Hollandric genes. The traits associated are called as Hollandric traits, and hypertrichosis, excessive hair on the pinna, is the example. Okay, good. So finally, after so many sessions, we are over with all the genetic disorders. Okay. So we saw a lot of genetic disorders. We saw chromosomal disorders, Mendelian disorders. And I think just, what is the homework today? You just have to sit down and just write down this table without cheating. Okay, just write down which are the disorders that we have done, and also write, the names. For example, aneuploidy. Aneuploidy, we have seen sex chromosome, we have seen autosomes. Write down the examples: Turner syndrome, Klinefelter syndrome, Down syndrome. Try to do it, without seeing. If you cannot, then just go back to the videos and see it, how it is. And tomorrow I will give you a better table with all the examples written, so that you have it in one shot. But do the homework. That's why I did not include it today, because it will be nice if you do it yourself and then you remember everything. Okay, good. Then let's revise. We'll do some questions today and then we'll go and eat dinner. Ames question. 3 children in a family have blood types O, AB and B respectively. What are the genotypes of their parents? What is the right answer? The blood groups that are possible are O, AB and B. These are the blood groups for the children. Okay, so then you have to tell me what are the genotypes of their parents that are possible. So you have to first write down the genotype of O blood group, the genotype of AB blood group, which are very easy, genotype of B blood group. And then you will automatically get to know what are the genotypes of the parents. Okay, I got a lot of answers, very good. But in between, I also see some wrong answers. Okay. So yeah, luckily, hopefully the pen is working. Let's see. Yeah, okay. So O blood group is represented by small I, or it can be represented as IO. So, if you just start with the O blood group, you will get the answer. What do you mean by this? This allele came from the mother, this allele came from the father, which means both of them should have either IO or I. In this case, they are represented with small I. So, both the parents should definitely have this allele in their genotype. A lot of you I saw were answering D, or maybe that was the answer from the previous question. Okay, whatever. So you understand, right? This itself will solve all of your questions, because one allele will come from the mother, one will be the father. So both the parents should definitely have small I, and then the only possibility is option A. And when you do the cross of these, you will observe that you also get B blood group, you also get AB blood group, you will also get A blood group. You will get all the 4 blood groups. Therefore, the right answer is option A. So, we will try to proceed. To the next question. So, everybody got right why the answer is A. And I am just blabbering because there are some technical issues here and there. But it will be fine soon. Good. So, The right answer is A. Next question. Given below is a highly simplified representation of human sex chromosome from a karyotype. And if you remember, I told you what is a karyotype. It is when you see the sequence of all the chromosomes. You remember the karyogram. Even if you don't remember that, just try to answer this AIMS question. So it is a highly simplified representation of human sex chromosome. What does gene A and B represent? It is again, those of tricky questions. But try to give me the right answer. Just write, 2. Just write 2 and then write your answer. Read the question properly. Read. Also, when I gave my exam, it was paper based, and I would always, when I read the question, I would underline the important things. For example, here I will definitely underline human sex chromosome. So then it is easy to answer. If you don't know the answer, just write don't know. That is okay. It's completely fine. Good. I start getting some answers. Good. Perfect. All of you have answered Haemophilia and red-green color blindness, and that is the right answer. Very good, guys. See AIMS question. And I know this is because we discussed, I asked you where are the genes for G6PD present. So that is why, just try to be very vigilant while you answer the questions. Good. So the right answer here is option number C. Haemophilia and red-green blindness. Everybody answered correct. Good. Next question. Mother and father both have blood group A. They have 2 children, one with blood group O and second with blood group A. They have, and then they give you 4 possible genotypes of the parents. What is the right answer? Another AIMS question. Over the course of these 7 sessions, we have already discussed more than half of the previous year AIMS questions. In the portal, I will also upload all the questions, so that you can practice all of them. And we have practiced, I think, around 50 to 60 questions in all these sessions, which is a very good thing. So, what is the answer for question number 3? Mother and father both have blood group A. They have 2 children, one with blood group O, second with blood group A. What are the genotypes? Good, good. I start getting the answers. Good. So, the right answer is option number D. Both are heterozygotic. Good. Then let's proceed. Did everybody get it? I hope everybody got it why it is heterozygotic, because there is O blood group involved, right? So they will definitely have one IO or small I gene, right? And that is why it is heterozygous. Good. So assertion and reasoning time. What is the right answer? The assertion is Haemophilia is a recessive sex-linked disease. The reason is haemophilia occurs due to mutation of a structural gene on chromosome 15. Again, for these questions, you have to write down if it is true, assertion is true, reason is true, if reason is the right explanation for assertion or not. We saw a very similar question even in our last lecture with phenylketonuria. That is why I am always saying chromosome, these are very important that you should know what is dominant, what is recessive, where are the genes present. I start getting some answers, but I am waiting till I get answers from everybody. Very good. So everybody answered option number C. which is the right answer. Yeah, see, you see for yourself, no, how slowly and slowly you are improving every day. You know, you're trying to solve so many questions and you're getting the right answers. So hemophilia is a sex-linked disease, but the genes are present on, write down in the chat box. I mean, I know all of you know the answer, but I just want you to write it down. So where are the genes for hemophilia present? Just write it down in the chat box. Everybody who answered C, because they already know where the genes are present, that is why they know reason is incorrect. So where are the genes present? Which chromosome? Just write it down quickly in the chat box and then we go have dinner. Good. I start getting the answers, you see. So just remember, okay, that is why, you know, in every session we have taken a different topic. Just try to remember what we did in this session, what we did in that session, and then it will be perfect. Very good. So the genes for hemophilia are present on the X chromosome or on the sex chromosome. Okay, that is why this is wrong. Perfect. So homework for today is that you have to write down yourself on the sheet of paper next to you all the disorders. Just write down their names, okay? Like we have seen this table, right? Chromosomal disorders, Mendelian disorders, polygenic, monogenic, you know, all these. And then also write the examples: Turner syndrome, Down syndrome, Klinefelter syndrome, ABO blood group, wherever it is, write. Okay, just write it down. Keep practicing questions, okay? And tomorrow we will study pedigree analysis. Okay, good then. Have a nice evening. I really hope you enjoyed the sessions despite the technical errors. We'll also repeat the question that was not very clear or that was a bit messy today. Okay, bye.